thank you for coming to my talk. My system is a little bit different. We've been hearing a lot about hot spring systems, and so this is a terrestrial weathering system. So I would just like to start out by framing this in an astrobiological context. So this classic paper here basically did free energy calculations for the weathering of iron-bearing silicate minerals, such as phaolite or ferrosilicate, and you actually could have, based on just the free energy of these reactions alone, produced an estimated 20 grams per cubic centimeter of biota over Martian history. And while that is less than you would predict based on for phototrophy, it's also not entirely inconsequential. So press it like you mean it, they weren't lying. So we also know that microbes are able to directly access the iron that is bound in the silicate mineral lattice for an energy metabolism. And this was demonstrated very beautifully by my advisor's lab group prior to my arrival at UW. And what they showed was the microbial lithotrophic oxidation of the structural iron in biotite. So that was by tracking the ingrowth of cells coupled to the oxidation of iron, as well as consumption. This was done under nitrate-reducing conditions. So this is also coupled to the decrease in nitrate. But what was really elegant about this study was the actual demonstration of what's going on with the mineralogy. So there was also all this beautiful microscopy and spectroscopy that went into this. And one of the interesting things that you see is actually the expulsion of iron oxide nanoparticles from the biotite structure. And that could be a result of the charge imbalance and then subsequent expulsion of that. So putting all of those things together, the proposed mechanism or proposed model of this was a direct enzymatic attack on the mineral surface. And that's in contrast to a abiotic dissolution process whereby you would have an iron moving into solution and then subsequent utilization of that. So this was proposed to be a direct enzymatic attack mediated by, wrong button, mediated by outer membrane proteins. And we will get into those a little bit later. So I wanted to take this to the field. I wanted to find an environment, perhaps, where a process like this would be relevant. So humor me for just 1 second. I am not about to tell you that I think Mars is made of granodiorite. However, this is the Rio Blanco quartz diorite of the Luquillo Mountains in Puerto Rico. So this is a extremely rapidly weathering granitic system. But what we have is appreciable weight percents of the iron-bearing minerals biotite and hornblende. And so this is a picture of it that was exposed by a landslide. And you see this beautiful spheroidal weathering pattern that is very typical for a granite. And what's driving this, coincidentally enough, this weathering pattern is actually the first step in the weathering is the oxidation of the structural iron in the mineral biotite. Convenient. So this is weathering very, very rapidly, even for a granitic system. And so there's also evidence based on this paper from Heather Buss, 2005, et al., I'm sorry, the rest of you, that there may be lithotrophy involved here. I don't have time to go over it, but there's a really beautiful reactive transport model coupling the availability of iron to the ingrowth of cells. But what's interesting to note here is that where you have high concentrations of extractable ferrous iron, you also have high concentrations of cells based on both direct counts and extractable DNA. So we set out, so conceptually, you can think about it like this. So here is a core stone that's been exposed by a road cut. And if you know anything about geologists, it's that we love road cuts. Because otherwise, you wouldn't be able to see something like this. So this conceptual model would actually be taking place at between 5 to 8 meters depth. And so you have unaltered bedrock, which looks like this. And on top of that, you have that kind of onion skin peeling structure termed the Rhineland zone. And that's where all of your plagioclases and your the hornblende weathers out and does not persist in the overlying saprolite. And the saprolite is variable in thickness here between 5 and 8 meters. So the idea is you have a heterotrophically dominated community at depth in your surface soil, and then you have where those high cell densities are, again, a chemolithoautotrophically dominated community that is driven by the oxidation of iron in the silicate mineral in interaction with a direct outer membrane protein, or this direct enzymatic attack. So this is the idea that through this process, these microbes are playing a direct role in the conversion of rock to regolith. Okay. So in June of last year, I went to Puerto Rico and did some sampling. We hand augured 3 cores to the depth of auger refusal, which was taken to be the bedrock saprolite interface. And the deepest point was 785 centimeters. And we collected samples down the saprolite profile for microbiological and genomic analyses. And this is, how it looks like, or what it looks like. All right. So just to kind of retrace what Heather did, I actually wanted to know, are these cells active, right? Do we just have dead, dormant cells accumulating at the bedrock saprolite interface? And to answer that question, then we did ATP bioluminescence assay, measuring the ATP. And if you remember anything from high school biology, it's probably that ATP is the energy currency of the cell. So I think this is a beautiful diagram where you have high concentrations of Fe total, iron total, and that's by dilute HCl extract. The ATP tends to track that very, very nicely. So this is great to see. Not only do we have, increased cells just by direct counts, but these cells actually appear to be active. I also established enrichment cultures for these using the whole rock granodiorite, or the constitutive minerals. Did it again. And again, called that one. So hornblende, biotite, and then also the other major phases being albite and quartz. So, so they got rock, they got groundwater, and they got carbon dioxide. And then we track them over time. And for simplicity and for brevity, I will only talk about the diorite enrichment cultures, because this is home cooking to these microbes. It's their actual native environment. So what you see with time is so we're out to 172 days. These are still ongoing. They're not done yet. I should probably sample them again. Is that in two of the sets, A and B, so the A, B, and C correspond to the material collected from the bottom of the 3 separate holes, it appears that there is actually oxidation as measured by the Fe2 to Fe total ratio. And then there also appears to be the generation of ATP in these cell cultures. So it looks like you have the generation of ATP coupled to the oxidation of iron in the rock. So this, now moving on into a little bit of metagenomics. So you may be tired of hearing about these systems already. However, these are really important systems for lithotrophy, these extracellular electron transport reactions. So in the last talk, Nathan spoke about more about this type of system, the porin cytochrome system. Whereas I'm going to talk or focus more on these SciC2 type systems, because that's what seems to be abundant here. So the model organism for this is Acetithiobacillus ferrooxidans. And what you have is the membrane-soluble protein that then is embedded itself, no need for an extra porin. And you have, that oxidation of the iron, and then the electrons are transferred to a periplasmic, either another cytochrome or or some other electron carrier in the periplasm. So in my metagenomic analysis, I found SciC2 homologs, several of them. And so this is 7 of the top 12 taxa actually have a homolog to SciC2. And so up top, these are gene maps. The model organism, Acetithiobacillus ferrooxidans, and the brown is the actual SciC2. And on the N terminus here is the number of heme binding motifs. And on the C terminus here is the number of transmembrane domains. And in Acetithiobacillus ferrooxidans, you see it's 1 in 18. And then next to that, you will find apparently I'm missing a number. There's a heme binding motif in this periplasmic cytochrome. So next to that, you have a periplasmic cytochrome to receive electrons from the Sci-C2. And so all of these first 5 here, are beta proteobacteria. These are not particularly canonical iron oxidizers. However, it's not entirely unreasonable to expect something like this, particularly in a Cupria vitis. There's known iron oxidizing Cupria vitis taxa. What's actually interesting to me here is these Xymanthomonaceae. They're gamma proteobacteria. And in this case, it has the periplasmic cytochrome, but it also, in another case, has rather than a cytochrome, a high potential iron sulfur cluster. And we also see that in one of these Cupria vitis strains. So to quote my advisor, to quote Nathan, who quoted my advisor, when you see Rubisco and EET together in one metagenomic bin, it's kind of a smoking gun for lithoautotrophy. And that appears to be what we have here. So just to wrap this up, I would like to revisit this conceptual model that I laid out earlier. So we have the Rio Blanco quartz diorite. On top of that, we have the saprolite. And we've demonstrated that not only are there cells there, but they're also active, right? So this is a good news. We have an active microbial community in situ. And then also, in enrichment culture, we have demonstrated that there is an apparent coupling of the oxidation of iron to the generation of ATP. But then the smoking gun in all of this was that interaction with these outer membrane proteins. And, well, there you go. We have Sci-C2 homologs in a fairly considerable number of taxa within these cultures. So it does appear that this idea that rock to regolith is catalyzed by bacteria does actually hold. So I would just like to thank my collaborators, as well as the U.S. Forest Service and the Luquillo Critical Zone Observatory for logistical help sampling these. And then my funding sources, the NEI and Department of Geoscience at UW-Madison. And I have time for some questions.
Thanks, Stephanie. Do we have any questions? So I've got one.
Yes.
So at what point in that sediment column does it go anoxic? And do you find evidence in your...
It doesn't.
It does not.
No, it's above the water table.
Okay.
So this is up on a ridge. It's not even suboxic.
Okay.
Yep.
Any additional questions? Okay, so again, thank you, Stephanie.